The digestive system begins in our oral cavity. The oral cavity is under the voluntary control of skeletal muscle. Through movement of the tongue and jaw, we are able to accomplish chewing. Chewing allows us to break down and grind our food into smaller pieces in order to facilitate swallowing. It also allows more of our food to be exposed to our salivatory enzymes, which begin digestion. This mixes the food with the saliva and stimulates our taste buds in order to give eating a pleasurable sensation. From the mouth, food is moved into the pharynx. The pharynx is a common pathway between the nasal cavity and the oral cavity and is a shared part of both the respiratory and digestive tract. From the pharynx, we see a splitting between the esophagus and the larynx. The larynx moves air into the lungs. The esophagus allows food to move down into our stomach. The ability to close off the larynx while swallowing is key to preventing respiratory infection. Food is prevented from entering into the trachea primarily by the elevation of the larynx and the closure of vocal folds across the laryngeal opening and sealing off the glottis entrance. The bolus tilts a small flap known as the epiglottis backwards and over the closed glottis to protect from food entering into the respiratory airways. Both ends of the esophagus are guarded by sphincters. A sphincter is a ring-like muscular structure that, when contracted, closes off the tube and prevents passage through. In the upper esophagus is the pharyngeal esophageal sphincter. Except for during swallowing, this sphincter is closed and prevents large volumes of air from entering into the esophagus and stomach during respiration. Once swallowing is initiated, a peristaltic wave sweeps from the beginning of the esophagus, forcing the bolus of food further downward into the stomach. This contraction moves progressively forward, pushing the bolus into the relaxed area ahead of the contraction. The peristaltic wave takes about 5 to 9 seconds to reach the lower end of the esophagus. From the esophagus, the bolus of food enters into the stomach, which is a J-shaped chamber between the esophagus and the small intestine. The entire time that it takes a food bolus to enter from the esophagus to the stomach is 6 to 10 seconds. The stomach can be divided into 3 distinct sections. The upper portion of the stomach is known as the fundus, connecting the esophagus and the stomach. The largest part of the stomach is the body, and the antrum, which connects the stomach to the small intestine. Connection between the esophagus and the stomach can be closed off by the gastroesophageal sphincter. This barrier keeps the stomach and the esophagus from interaction, which is particularly important because of the acidic gastric juices within the stomach. If the gastric contents did move backwards up the esophagus, acidity of these contents would cause irritation to the cells within the esophagus. The end of the stomach is separated from the small intestine by the pyloric sphincter. This acts as a barrier between the stomach and the upper part of the small intestine. When emptied, the stomach has a volume of about 50 milliliters, but can expand to hold approximately 1 liter of food. It can accommodate this change with little change in tension or pressure due to the deep folds found in the stomach when emptied. As the stomach fills, these folds get smaller and flatten out as the stomach relaxes slightly with each amount of food that comes into the stomach. This ability to increase in volume allows the stomach to perform its most important function of storage of ingested food until it can be emptied into the small intestine at an appropriate rate for digestion and absorption to occur. The stomach also continues to mix ingested food with gastric secretions to produce a thick liquid mixture known as chyme. The stomach contents must be converted into chyme before they can be entered into the small intestine. Chyme is produced by the peristaltic contractions within the stomach. A group of pacemaker cells located in the upper fundus region generate slow wave potentials that sweep down the entire length of the stomach towards the pyloric sphincter. These contractions spread over the fundus and travel down the body and into the antrum, where they become more vigorous because the antrum has a thicker layer of muscular tissue. Initially, as the chyme is moved closer to the pyloric sphincter, the pressure the chyme exerts on the pyloric sphincter completely shuts it. A small amount of water and other juices are able to briefly escape into the small intestine prior to the chyme fully closing the pyloric sphincter. With each wave, only a few milliliters are able to move through the pyloric sphincter. The remainder is pushed back upwards into the antrum, causing further mixing of the chyme within the stomach. The chyme will remain in this location until the next wave of contraction moves through the stomach. In addition to the mixing that happens in the antrum, these peristaltic contractions are the driving force for gastric emptying. The amount of chyme that escapes into the small intestine before the pyloric sphincter is closed is largely dependent on the strength of the contraction. These contractions can vary greatly in intensity based on several factors. The four most important factors that influence gastric emptying are fat, acidity, hypertonicity, and dissension in the small intestine. Because the small intestine is the main location of digestion and absorption, anything that would hinder these two processes is going to decrease the rate at which the stomach releases content into the small intestine. Fat takes longer to digest and absorb than other nutrients. When there is a lot of fat already in the small intestine, further gastric emptying of more fatty contents is prevented until the small intestine has already processed the fat within it. The presence of fat is the most potent stimulus for inhibiting gastric motility. The acidity within the small intestine is tightly monitored. This is because unneutralized acids irritate the mucosa of the small intestine and inactivate pancreatic digestive enzymes. Chyme has a high acidity due to the hydrochloric acid it secretes. If too much chyme enters into the small intestine, this decreases the ability of the small intestine to neutralize it and will lead to an increase in acidity. This will slow gastric emptying by decreasing the intensity of antrum contractions until the small intestine is able to neutralize the acids present within it. Osmolarity depends on the number of molecules present, not the size. Even though one protein molecule may be split into several hundred amino acids, each of which has the same osmolarity effect as the original protein molecule. This is true for starch and fats. Because water freely moves between the small intestine and the plasma, If osmolarity increases within the small intestine, more water will enter into the small intestine to dilute its contents. This becomes problematic for the cardiovascular system, as too much plasma diffusing from the capillaries into the small intestine will reduce plasma volume and decrease stroke volume. To prevent too much plasma from entering into the small intestine from the plasma, gastric emptying is inhibited when the osmolarity of the small intestine starts to rise. This prevents further food molecules from entering into the small intestine and maintaining proper osmolarity. The last stimulus for a decrease in contraction within the antrum or a decrease in gastric emptying is dissension. If too much chyme or too much water from too much hypertonicity is present in the small intestine, this inhibits gastric emptying to prevent further distension. Beyond the stomach, chyme enters into the small intestine. The small intestine is responsible for the remainder of digestion and the majority of absorption. The small intestine is responsible for absorbing all nutrients and the majority of all other constituents. This large intestine does do some absorption of water and sodium, but nothing in comparison to the small intestine. The small intestine is a long tube that's coiled underneath the stomach within the abdominal cavity. The small intestine, like the stomach, is arbitrarily split into 3 segments. The duodenum is right beyond the pyloric sphincter, followed by the jejunum, which is the central part of the small intestine, and the portion of the intestine closest to the large intestine is the ileum. In the stomach, contraction initiated by pacemaker cells in the fundus continuously spread throughout the stomach, ending by the pyloric sphincter. This allows a small amount of mixing to occur within the antrum of the stomach and also forces food down towards the antrum. In the small intestine, contraction is by area. It does not continuously spread from the top of the small intestine down to the bottom of the ileum. Instead, what happens is an alternating contraction by area. An example of this would be the purple arrows represent the area that is contracting, and the orange arrows represent the area that would be in relaxation. A contraction would occur in one segment, and the segment just beyond that would be going through relaxation. This allows the forward propulsion of the chyme through the small intestine. After a brief contraction period, that area would then enter into relaxation, while the area that was just in relaxation would enter into a contraction phase. The first contraction of the small intestine causes those areas of contraction to force chyme both forwards and backwards into the relaxed areas of the small intestine. After the first contraction, the small intestine segments under relaxation are partially filled with chyme from the small intestine segment just before it and just after it. As the second wave of contraction would begin, this would cause the mixing of the previous segment's chyme and the forward segment's chyme, which allows the chymes to fully integrate and exposes more of the chyme to the digestive juices and enzymes of the small intestine. Even though the movement of chyme is both forwards and backwards with every contraction, propulsion still occurs. Propulsion is accomplished by varying frequencies of contraction and varying size of segmentation throughout the small intestine. In the duodenum, contractions occur at a rate of approximately 12 contractions per minute. This is a faster rate than what occurs in the ileum, where the rate of contraction is only 9 per minute. This means that more chyme is moved and contracted in a forward motion early in the small intestine compared to later in the small intestine. This more frequent contraction forces more movement of chyme from the top of the small intestine towards the end of the small intestine. The small intestine also varies in the size of segmentation from the duodenum to the ileum. Segmentation is more frequent in the duodenum, and therefore the size of the segments are smaller. As you move through the jejunum and the ileum, the segments become larger in size. This means they can hold more chyme before they are filling compared to those lying before it. This forces more chyme forward into the small intestine, moving towards the large intestine, then backwards, where the segment fills more quickly. After the majority of the meal has been absorbed, the segmentation contractions cease and are replaced by a migration motility complex. This is a contraction that starts in the stomach and migrates down throughout the small intestine. Each new wave is initiated a little further down the small intestine and helps to remove remaining constituents plus the mucosa debris and bacteria towards the colon. Once this wave has reached the end of the small intestine, the process begins again, which allows the small intestine to continually cleanse itself regardless of if food is present. The ileocecal valve is surrounded by the smooth muscle of the ileocecal sphincter. The valve acts in a one-way fashion, allowing content from the small intestine, when pushed against the valve, to force it open. However, the ileocecal sphincter is able to decrease or increase the amount of space the ileocecal valve opens by its rate of contraction. Greater contraction results in a smaller opening, while a decrease in contraction results in a greater opening. As the content of the cecum increases, you can see that the backwards flow of this content onto the ileocecal valve forces it shut. Again, the ileocecal sphincter can decrease the opening of the ileocecal valve by increasing its contraction, which occurs when content in the cecum increases the pressure it exerts on the cecal valve. This one-way valve system prevents the backflow of content from the large intestine back into the small intestine, and also keeps the large community of bacteria housed in the large intestine. If this bacteria were allowed to move from the large intestine into the small intestine, it would quickly multiply due to the rich nutrient-based content of the small intestine and could wreak havoc on our digestive system. The large intestine is composed of the colon, the sigmoid colon, the rectum, and the anal canal. The colon is the bulk of the large intestine and can be divided into the ascending colon, the transverse colon, and the descending colon. The S-shaped portion of the large intestine is the sigmoid colon. This connects to the rectum, which connects to the anal canal. The appendix is attached to the ascending colon. Each day, the large intestine normally receives about 500 milliliters of chyme from the small intestine. The main role of the large intestine is to hold these components and extract more water and salt from its content. After salt and water have been eliminated from the chyme, it is known as feces. Feces is largely composed of undigestible food such as cellulose, unabsorbed biliary components, and remaining fluid that was unable to be extracted. Cellulose and other undigestible substances provide the bulk and help maintain regular bowel movement. Halstra contractions usually occur once every 30 minutes. These contractions are similar to the contractions of the small intestine, where one segment is relaxing while the next segment is contracting. This slowly shuffles the content back and forth. These movements slowly mix its content back and forth, exposing more of it to the absorption mucosa within the large intestine. Movement in the colon is largely non-propulsive. These contractions are called Halstra contractions, and they're initiated by autorhythmic cells of the smooth muscle, which are largely controlled by the intrinsic plexus of the digestive system. The large intestine is segmented into small sacs similar to the small intestine. These sacs are known as halstra. They are larger and occur less frequently than those found in the small intestine. 3 or 4 times a day, generally after meals, there is a marked increase in motility that takes place within the transverse and ascending colon. A mass movement drives the content within the colon forward approximately 1/3 to 3/4 the length of the colon within a few seconds. This mass movement involves the simultaneous contraction of the ascending and transverse colon. This drives the content into the distal part of the large intestine, where material is stored until defecation occurs. Mass movements are triggered by the gastrocolic reflex. This reflex occurs when a new meal enters into the digestive tract and initiates the movement of existing content further along the tract to make way for the new incoming food. When feces move into the rectum, dissension of the rectum stimulates the stretch receptors within the rectal wall. This initiates the defecation reflex. This reflex causes the internal anal sphincter, which is a smooth muscle, to relax and the rectum and the sigmoid column to contract more vigorously. If the external anal sphincter, which is under skeletal control, is also relaxed, defecation will occur. Because the external anal sphincter is in skeletal muscle, it means that it is under voluntary control. If it is not an appropriate time to defecate, there is a voluntary tightening of the external anal sphincter, which prevents defecation despite the reflex. If defecation is delayed, the descended rectal wall gradually relaxes and the urge to defecate subsides until the next mass movement propels more feces into the rectum. During periods of inactivity, both the anal sphincters remain contracted